We live in an era of tremendous opportunities and also unprecedented challenges. One of the primary challenges is to sustainably produce more nutritionally dense food with the fewer resources to feed the growing world population. It is also one of the top United Nations Sustainable Development Goals and the research priorities in the Clemson Forward Plan. At PD Research and Education Center, the new breeding technologies are applied toward this goal, precisely to develop safe crops with sustained yields and nucleic acid-based pesticides to reduce reliance on non-environment friendly synthetic pesticides. Weed-caused diseases are quite common in the United States, with a prevalence of 7 to 10 individuals in every 100. Celiac disease can be prevented by avoiding gluten-containing food, but wheat avoidance has been shown to have unwanted health impacts in long term. Similarly, about 1.5% of Americans, including children and adults, suffer from peanut allergy. Avoiding peanuts is the only effective therapy, but due to ubiquity of peanuts' accidental exposures, due to changes in climatic conditions conducive to insect and fungal pest occurrence, the demand for pesticides has continued to increase worldwide. As conventional insecticides cause environmental pollution, there is a dire need to develop environment-friendly alternatives. Cotton yields are stagnant despite several attempts since the last decade. Also, the cultivated cotton is perennial by nature, however, we treat it as an annual. This change in treatment require applying chemicals like defoliants and desiccants to facilitate harvest and reduce regeneration. It creates a need to develop cotton genotypes with annual growth habit and enhanced yield with fewer input requirements to meet the growing demand for natural fiber. Keeping in mind these challenges, we undertook the following research objectives. Number one, develop wheat genotypes suitable for gluten sensitive and allergic individuals and peanut genotypes for individuals with mild peanut allergy. Number two, develop environmentally benign pesticides for sugarcane aphid that cause significant losses in sorghum, gray mold, and honey fungus, which cause considerable losses in peach, and aspergillus species in peanut, which contaminate grains with aflatoxins. Number three, develop cotton genotypes with annual or near annual growth habit with expected yield gain. To achieve the first objective, we undertook two approaches. Number one, profiling the seed storage proteins dubbed gluten from wheat landraces to identify genotypes lacking specific proteins and cross them genetically. Number two, use the CRISPR technology to induce mutations in the genes coding immunogenic gluten proteins. A similar approach is adopted in peanuts to develop genotypes with reduced allergenicity, except in this case we screened a collection of diverse peanut lines from the US Department of Agriculture. To develop environmentally friendly pesticides, we targeted the vital pest genes for suppression by topical application of RNA molecules. When absorbed by the pest from the host plant, these molecules cause silencing of essential pest genes leading to mortality. These molecules are highly specific and does not affect the organisms other than belonging to the target species. Moreover, these molecules do not have residual activity due to short lifespan in the environment. We are working to make these compounds affordable, finding chemical formulations that make them persist in the field conditions for the desired duration. For the third project, we measured the expression of the five essential cotton genes that integrate environmental signals and determine which bud should turn into branch or bear flowers and when in a collection of highly diverse upland cotton lines. The idea is to identify high expression variants of these genes and genetically cross genotypes bearing these variants to stack them in a single genotype. The expected outcomes are the development of wheat genotypes suitable for wheat allergic and gluten sensitive individuals, and peanut lines for individuals with peanut allergy. Develop eco friendly RNA based pest control compounds for sugarcane aphid, gray mold, honey fungus, and aspergillus species, and annual cotton genotypes with high fiber yield.